Hey folks, it's Steve from biology class. I figured it might be helpful if I went through some of the quizzes that we have, because there's a lab quiz for some of you tomorrow and some Thursday. We weren't fortunate enough to think about asking for a cheat sheet on Tuesday, so I figured I'd do this for the two folks on Tuesday. Whoever wants to use it can. I only have about half the quizzes, but I figure it's better than nothing. And they're in no particular order, so feel free to pause, rewind, whatever. I'll do my best not to murder it. First, I guess we'll go through one of the first ones because this is cumulative. And I think, where is he? Well, we got number ones in here somewhere. We have, yes. Well, I don't know if it's quiz number one, but it's a quiz for lab number 4, and that's cell dynamics, diffusion, and osmosis. And number 1 was, what is Brownian motion? You guys might remember. It's the random movement of very small particles in response to the movement of electrons. That's Brownian motion. What is diffusion? Diffusion is the non-random movement of a substance from a higher gradient of concentration to a lower gradient. And that's diffusion. What is osmosis? Osmosis is a special type of diffusion of water passing through a semi-permeable membrane to an area of higher solute concentration. And then the rest of that is number 4 and number 5 are drawings. It's just displaying, the direction of the solute and the water will go to the, I don't think that's relevant or helpful, but there you go. There's that. The arrows tell you everything. Okay, on to the next one. This is on DNA, and replication is the duplication of DNA. Transcription is the making of RNA, and translation is making a protein. Or information makes a protein. And then how many bases does it take to code for an RNA, excuse me, I'm dyslexic, codon. And for a codon, it is 3 bases. We all should know that. It's for 1 amino acid. And that's the next question. What does a RNA codon code for? And that is 1 amino acid. Chromosomes 1 protein. Codon's the amino acid. Good to remember those. And then I'm sure you guys can remember from translation from C pairs to G in DNA, and then A to T in DNA, and then it's A to U in RNA. Good to remember that difference. T does not exist in DNA, and U does not exist in RNA. So there's translations from for DNA it's A to T, and for RNA it's A to U. Helpful to remember those things. And then the 3 components of a DNA nucleotide are the phosphate, instead, the 5-carbon deoxyribose sugar, and then the nitrogenous base. Those are the 3 components of a DNA nucleotide. And then we have, well, this was a sketch, but it told you to, look at the human cheek cell and identify the nucleus, the cytoplasm, and the cell membrane. Obviously, we all know the nucleus was the tiny thing in the middle. The cytoplasm was the stuff in the middle, and then the cell membrane was, well, we know, phospholipid bilayer membrane. Anyways, it looks like that. Like that. And I'm sure you remember. Anyway, number 2. Why did we stain our slide prep of the cheek cell before viewing it through the microscope? Oh yeah, this is on microscope and cells. Well, that's so we could see the cell. It's different parts of the cell, so we stained it so it become more visible. That's why it wasn't obvious. Number 3. We saw the greatest detail of the stained cheek cell at high power. What magnification is high power? It's 4 times 400. 400 times magnification. Okay, if the width of your cheek cell is about 1/10 the width of the field of vision at 400 times the magnification, which is 350 nanometers across, what is the width of the cell? So obviously you're going to say if it's 1/10 of 350 nanometers, it's going to be 35, obviously. Pretty simple math. What is a scale bar? A scale bar is a tool used to show the difference in size relation relative to its scale. And you know, there are 4 different types of lenses and magnification times 10, which is times 4, which is times 40. Is scanning power. That's just 40 times magnified. And then the next one is 10 times 10. And then after that, that's low power. So that's 100 times total magnification is low power. And then 400 times, which is high power magnification. And then we have 1,000 times, which we weren't actually able to view, but that's oil immersion. So you have times 40 is scanning power, times 100 is low power, times 400 is high power, and times 1,000 is oil immersion. And then I'm sure you guys can remember everything about the Elodea leaf. I think that's how you say it. We have the cell wall, the cytoplasm, the chloroplast, and the central vacuole. Don't know how helpful that is. Obviously, I wrote organelles. That is incorrect, but if you can see that, that's how that works. Anyway, somehow it seems it's all backwards. Neat. Anyway, that's that. And then on the back, we have number 8. Cell theory states that all life is composed of cells. Cells are fundamental units which possess all characteristics of living things. New cells can only come into existence by the division of previously existing cells. This is true. The other ones were asking you to do math. There was a little bit of discrepancy here. I hope you guys just remember how to do the conversion. I was going to say, I think that there was some kind of mistake on that one. So I want to just warn you about that before putting too much faith into it. Okay. And number 10, looking through the ocular lens of a light microscope, object will be seen inverted, flipped, and upside down, and greatly magnified. Go on to the next one. This is one of the long quiz that we had as 20 questions. So this was on mitosis and meiosis. Okay, number 1, mitosis versus meiosis. In each case, the original cell is diploid. That is true. Number 2, referring to the number of complete sets of chromosomes in a cell, N is B, the number of actual chromosomes, duplicated or not, in that cell. Number 3, 1N equals the haploid condition. It is where there is but one complete set of chromosomes in that cell. So Haploid means one set of chromosomes. Number 4 is 2n equals the diploid condition. It is where there are 2 complete sets of chromosomes in that cell. And number 5, mitosis begins with a diploid cell. Number 6, mitosis is vital to an organism as this is how a cell makes new cells for growth, replaces cells in tissue, and it's how it repairs and replaces damaged tissues. So those are all important. So that's why mitosis is vital to an organism, because it's how cell makes new cells for growth, it's how it replaces the cells in tissues, and it's also how it repairs and replaces damaged cell tissue. Number 7. The cells produced by mitosis are genetically identical to the cell from which they are formed. This is true. They're identical. And these are the mitotic phases. Number 8 is the mitonic phases. And that is prophase, metaphase, anaphase, telophase, or telophase, whichever you want to say. And then it is interphase, which is in parentheses. So that's PMAT and then interphase. If that helps you remember, it helps me. Number 9. DNA duplication occurs in the S phase of interphase before prophase begins. That is also true. Number 10. By the time prophase begins in mitosis, the DNA is already duplicated. That is true. Number 11. In prophase of mitosis, duplicated DNA strands of chromatin are condensed to the duplicated chromosome form, with 2 sister chromatids attached to the centromere. Number 12. DNA condensed to chromosomes is achieved by winding around histone proteins. Number 13. Meiosis begins with a diploid cell. That is also true. Both mitosis and meiosis begin with diploid cells, not haploid. Meiosis will eventually form haploid cells for them. Number 14. By the time prophase 1 begins in meiosis, the DNA is already duplicated. I think I just said that. That is true. Number 15. Meiosis 1 is a reduction division producing 2 diploid cells. Oh, wait, that is not true. Producing 2 haploid cells. I misspoke there. 2 haploid cells. Ooh, careful. Meiosis 1 is a reduction division producing 2 haploid cells with 1 set of chromosomes each. And then number 16. In prophase 1, the homologous duplicated chromosomes are attached to each other to form tetrads. I'm going to read that again. Sorry for murdering it. In prophase 1, the homologous duplicated chromosomes are attached to each other to form tetrads. Still murdered it, but I think you got it that time. Okay. Number 17. Crossing over and the exchange of genetic information between homologous and chromosomes occur in metaphase 1. That is false. They occur in prophase. Crossovers occur in prophase 1. Number 18. The random alignment of tetrads along the central plane is called random assortment. That is not true. This occurs in both metaphase 1 and 2 and is the major source of change in the DNA contained in the resulting nuclei. No, this is false. The major change occurs from crossovers, as you know. Okay, number 19. During the prophase, reading the wrong one. The purpose of meiosis is to produce genetically different haploid gametes. That is true. Okay, number 20. The gametes are called egg and sperm. Well, yes, that's true. Oh. And, I just also realized I read you number 18 wrong. I'm going to read you number 18 again, because I don't want you to get mad at me for getting this one wrong. The random alignment of tetrads along the central plane of a cell is called random assortment. That's actually true. I got that wrong. I didn't notice. This occurs in both metaphase 1 and 2 and is a major source of change in the DNA contained in the resulting nuclei. I thought it was from. Apparently I can be wrong. We all can. But there you go. That is true. So remember that. And that's it for that one. And then we have this one, which is on heredity and epidemiology. And there's some terms. And number 1 tells you what a gene is. A gene, a discrete unit of hereditary information consisting of a specific nucleotide sequence in DNA. That's what a gene is. An allele, an alternate form of a gene. And then we have homozygous, which is having 2 identical alleles for a given gene. And then there's heterozygous, which is a cell with 2 different alleles for the same trait. And then number 5, dominant allele, which is in a heterozygous individual, the allele that determines the phenotype with respect to a particular gene. And then recessive allele is in a heterozygous individual, the allele that has no noticeable effect on the genotype, meaning the one that is not expressed. Phenotype is the last one, and that is C, physical appearance or the expression of that gene. Then the rest is going through different types of stuff there for, our little boxes we have to do, and then we're going to probably have to do a pedigree. I imagine we might have to do things like that, but that's about half of the quizzes that I had available. The ones that I can't find are probably stuff on, well, I would imagine the Krebs cycle, cellular respiration, stuff like that. pH, I didn't have that one. Stuff like that. You're going to want to remember what an acid is, what a base is. Like I said, stuff about the Krebs cycle, photosynthesis. Those are all things you're probably going to want to study. This is the second time I've done this video. The first one got messed up, but already I kind of remember some of this stuff better, so I hope it helps you. Study hard. I will see you guys when I see you, and do your own test. Have a great night.